welcome to the deep dive. Today, we're looking at something that, well, it seems almost counterintuitive at first glance. Immense biological power packed into something incredibly simple.
It really is fascinating. We're talking about minimalism in biology.
Exactly. We're focusing on one molecule, KPV. It sounds complex, maybe, but it's structurally about as simple as you can get, and yet the early research suggests it might be revolutionary for inflammation and repair.
Yeah, KPV stands for lysine, proline, valine. Just 3 amino acids. KPV, that's it, a tripeptide.
Three little building blocks, doing work that you'd normally think needs, huge proteins or complex signaling chains. So our deep dive today, it's based on, well, quite a stack of research papers, expert commentary on KPV, often called the anti-inflammatory repair peptide.
Right.
And our mission really is to unpack how this tiny thing works inside our cells, looked at the pretty impressive results from preclinical studies, and also, crucially, face the big hurdles, the translational challenges that need solving before this could ever reach, say, a clinic.
Absolutely. And understanding where it comes from, its origin story, that's actually key to why it's interesting.
Okay, so where does it come from? It's not just synthesized out of thin air.
No, not originally. It's actually a natural piece.
Yeah.
KPV corresponds exactly to the last 3 amino acids, the C-terminal end, of a much bigger, well-known hormone our bodies make.
Which one?
It's called amelanocyte-stimulating hormone, or MSH for short. KPV is literally the tail segment of AMLSH.
So it exists naturally inside us already as part of this larger hormone.
Interesting. So why the focus on just the tail? Why not use the whole MSH molecule if it's already there and known?
Well, that's the clever part. It's about getting rid of unwanted baggage. The parent molecule, MSH, it's great for anti-inflammatory effects, very potent, but it does a lot of other things too. It binds strongly to melanocortin receptors, MC1R especially, which triggers things like skin pigmentation, the tanning effect.
Right, the melanocyte-stimulating part of the name. Exactly. And potentially other systemic hormonal effects you might not want if you're just trying to target inflammation. KPV is kind of engineered by nature or selected by researchers because it keeps a lot of that anti-inflammatory power. But because it's so small and structurally different enough, it's much, much less likely to hit those pigment receptors or cause broad hormonal side effects.
So KPV is like the distilled essence, the core function, the anti-inflammatory bit, without all the extra noise. The business end, maybe?
That's a great way to put it. The active signature, stripped down. Optimized for information control.
Okay, this is fascinating. Let's get into the science then. How on earth does something so small manage to have such a big effect inside a cell?
Yeah, this part is really where the standard thinking gets challenged. It's the aha moment in the KPV story.
Because normally you think peptides, hormones, they need a receptor, right? Like a key fitting into a lock on the outside of the cell to send a signal inside. That's the classic model. Was that the first guess for KPV?
Absolutely, that was the logical assumption. Given it comes from MSH, everyone thought, okay, it must work through the same receptors, primarily those melanocortin receptors, MC1R, the ones involved in pigmentation.
Makes sense.
But then the studies came in and, well, plot twist, KPV's powerful anti-inflammatory effects, they seem to be largely independent of those MC1R receptors.
Independent. Wow. Okay, that changes everything for how you'd think about using it. So if it's not using that main doorway, what's the proof? How do we know it's non-melanotropic?
Well, there are several strong lines of evidence. First, researchers tried using antagonist molecules specifically designed to block or jam the MC1R receptor lock.
Right.
If KPV needed that lock, the blocker should stop it from working. But it didn't. KPV's anti-inflammatory action continued even when MC1R was blocked.
Okay, that's pretty compelling. What else?
Second, they used experimental models like cell lines or even animals that were genetically engineered to completely lack functional MC1R. They just didn't have the receptor.
And KPV still worked.
It still worked. It retained its full anti-inflammatory function. That's about as definitive as it gets if the target isn't there, but the effect still happens. It's not using that target.
Undeniable, really.
And the final piece is biochemical. Activating MC1R normally triggers a specific internal signal, a rise in something called intracellular CAMP. KPV doesn't do that. It doesn't cause that CAMP spike.
Got it.
So all that together really solidifies its classification. KPV is a non-melanotropic anti-inflammatory peptide, which is a huge plus if you want targeted effects without, say, unintended tanning.
Okay, so if it's not using the front door receptor, how is it working? Is it sneaking in somehow? Going straight to the, I don't know, the control panel inside the cell?
That's pretty much the picture that emerges. The evidence strongly points towards KPV directly influencing key pathways inside the cell. And this takes us to arguably the absolute master switch for inflammation.
Which is?
A protein complex called NFE, nuclear factor kappa B.
NFKA. Yeah, you hear about that one a lot. It's like the cell's big red alarm button, right? It tells the cell to panic and start pumping out inflammatory stuff.
That's a perfect analogy. Think of it as the central command for stress and defense. When trouble hits infection, injury, stress, NFK gets activated, moves into the nucleus, and switches on the genes for all those inflammatory molecules. It translates the external threat into an internal molecular response.
The inflammatory storm.
Exactly. And KPV seems to intervene right there at that central switch.
But how? If it's not using a receptor on the outside to relay the message, how does it physically get inside and affect NFK?
Right, that's the million-dollar question. The mechanism seems to involve modulating the molecules that control NFEB. Specifically, there's a key inhibitor protein called IB.
By kappa B alpha.
Okay. Normally, IB acts like a bodyguard. It binds to NFD and holds it captive, inactive, out in the main part of the cell, the cytoplasm.
Keeping it quiet?
Right. When an inflammatory signal arrives, the cell quickly destroys IBIS. This releases NFD, freeing it to rush into the nucleus and sound the alarm.
Okay.
What KPV appears to do is accelerate the recovery of that IBIT bodyguard. It helps the cell rebuild its inhibitor shield faster.
Ah, I see. So it's not just blocking the alarm signal itself, it's actively helping the cell put its own natural brake back on the system by restoring the inhibitor. That's elegant.
It's incredibly elegant, yes. It works with the cell's own regulatory system. By speeding up the synthesis and stabilization of IB, KPV helps turn that master inflammatory switch back down much more quickly.
And the result of that? Downstream.
The result is a significant drop in the production of those pro-inflammatory molecules, those cytokines like interleukin-8, IL-8, and it also seems to interfere with the signaling of another major inflammation driver, IL-1.
IL-1 beta, yeah, that's a big one in chronic inflammation.
Absolutely. So KPV hits a really fundamental control point.
Okay, the mechanism inside the cell is clearer now, but we still have the problem of getting it there. You mentioned it's small, which is good, but it's also hydrophilic. It likes water, right?
Correct.
Which usually means it's terrible at crossing cell membranes, which are fatty, lipid-based. So if you just injected KPV into the bloodstream, wouldn't most of it just wash away, unable to get into the cells where it needs to work?
That poor permeability, yes, that is a major, major roadblock for systemic use. It's small, but it can't easily slip through those fatty cell walls.
But you hinted there might be an exception, a specific place it can get in easily?
Yes. This is one of the really neat biological details. There's a sort of secret door, a specialized transport system that exists primarily in one place.
Yeah.
The lining of our gut. The intestines.
The gut has a secret door. Why the gut specifically? What is it?
The cells lining the intestines, the enterocytes, they have a high concentration of a special protein transporter embedded in their membranes. It's called PEPT1, peptide transporter 1.
PEPT1. And its job is?
Its natural job is to grab small peptides, dipeptides, and tripeptides, usually from digested food protein, and actively pull them from the gut contents into the intestinal cells and then often into the bloodstream.
Ah, so the body already has a system designed to absorb small peptides from the gut.
Exactly. And KPV, being a tripeptide, just happens to be the perfect size and shape to be recognized and transported by PEPT1. It fits the lock, so to speak.
So if you can get KPV to the gut, PEPT1 acts like a dedicated entry point.
Precisely. This explains why KPV shows such strong, localized effects in animal models of gut inflammation, like colitis. Oral delivery, or delivery systems targeting the colon, can leverage this built-in uptake mechanism.
That makes so much sense. But it also highlights the flip side, right? What about tissues that don't have much PEPT1, like the brain or joints?
Exactly. In those tissues, that easy access just isn't there. The secret door is closed. And that's where the delivery challenge becomes really critical and requires much more sophisticated approaches.
Okay. Understanding that PEPT1 transporter mechanism really sets the stage. It tells us why the gut is such a prime target for KPV research. Let's dig into that evidence now. What do the actual experiments show?
Well, as you'd expect, the most extensive work by far has been done in models of intestinal inflammation. Specifically, mouse models of colitis, which are designed to mimic aspects of human diseases like Crohn's or ulcerative colitis.
And how did they get the KPV to the inflamed area in these mice?
They used methods designed to take advantage of that PEP-T1 transporter, either simple oral administration, basically feeding it to the mice, or sometimes more advanced methods like loading KPV into nanoparticles.
Nanoparticles, why use those?
These can be engineered to protect the KPV from being degraded on its way through the stomach and small intestine, and then release it specifically in the colon, where the inflammation is worst, maximizing uptake via PEP-T1 there.
Clever.
Yeah.
So what did they actually measure? What were the results in these colitis models?
The results reported in these studies are pretty consistent and, frankly, quite impressive. They saw significant reductions in inflammatory cytokine levels, measuring that inflammatory storm we talked about.
Less inflammation signals.
Right. They also observed less physical tissue damage. The gut lining looked healthier under the microscope. And overall improvements in clinical signs of the disease in the animals, like weight loss or diarrhea.
And did they definitively link this benefit back to the PEP-T1 transporter?
Yes. Crucially, they did control experiments. In some studies, they used a chemical to specifically block the PEP-T1 transporter in the gut.
Okay.
When PEP-T1 was blocked, the beneficial effects of KPV were significantly reduced. That really confirms that PEP-T1 is the main gateway for KPV's action in the gut. It ties the mechanism directly to the positive outcome.
That's solid confirmation. But okay, let's broaden out. The research seems to suggest KPV does more than just, turn down the inflammation volume. Yeah.
Uh.
It acts more like like you called it, a molecular paramedic, helping with the cleanup and repair too.
That's right. And this dual function is really what makes it potentially more interesting than just a standard anti-inflammatory. It seems to promote what's called cytoprotection.
Protecting the cells themselves.
Exactly. Protecting the cells from the damaging side effects of the inflammation itself, like oxidative stress. Inflammation generates a lot of harmful reactive oxygen species, free radicals that damage cellular components. KPV seems to boost the cell's own antioxidant defenses, helping shield them.
Okay, so it calms the fire and protects the building from smoke damage, so to speak.
Hmm.
What about the immune cells involved? Does it just shut them down?
No, it appears to guide their behavior more constructively. There's evidence KPV influences macrophage polarization. Macrophages are those big immune cells, kind of like the heavy lifters of the immune system.
Oh, yeah.
They can exist in different states. There's the M1 phenotype, which is very aggressive, pro-inflammatory, the attack mode. And then there's the M2 phenotype, which is more anti-inflammatory, focused on cleanup and repair. KPV seems to encourage macrophages to shift away from that aggressive M1 state towards the M2 repair-oriented state.
So it's not just hitting the brakes, it's actually shifting gears,
Yeah,
moving the immune response from attack towards heal.
that's a great way to think about it. It helps transition the local environment towards resolution and repair. And that's obviously crucial for healing damaged tissues like the gut lining and colitis or potentially wounds elsewhere.
This combination, turning down NFB and promoting repair phenotypes, sounds really powerful. And you said this protective action isn't just limited to the gut.
No, the potential seems broader, although delivery becomes the challenge again. But let's look at some specific examples from the research in other tissues.
Okay, where else has it shown promise? What about something really complex like the brain?
Yeah, this was quite striking. In animal models of traumatic brain injury, TBI.
Which involves massive inflammation.
Exactly. A single dose of KPV given after the injury showed some remarkable effects. It actually reduced the physical size of the damaged area, the lesion, and significantly decreased the number of neurons dying off.
It stopped neurons from dying. How?
It seems to be linked to both its anti-inflammatory effect and something called anti-apoptotic action.
Anti-apoptotic.
Yeah.
Meaning it prevents programmed cell death. Apoptosis is like cell suicide, right?
Correct. After brain injury, a lot of neurons don't die immediately from the impact itself. They die later through this programmed self-destruction pathway, triggered by the toxic inflammatory environment. KPV seems to help stabilize things and interfere with those self-destruct signals.
So it offers protection beyond just reducing the initial swelling or inflammation. It helps prevent that delayed wave of cell loss.
That appears to be the case in these models, yes, offering a window for neuroprotection.
Wow. Okay, what about other vulnerable tissues, like the lungs? We hear a lot about inflammation from air pollution, for instance.
Yes, there's research there too. In models simulating lung injury caused by things like fine dust particles mimicking occupational or environmental exposure, KPV administration reduced markers of oxidative stress in the lung tissue, lessened the visible damage, and cooled down the overall inflammatory response.
So potential applications there for chronic respiratory conditions driven by inflammation.
It's a possibility that researchers are exploring, yes.
Okay, and let's circle back to the skin. You mentioned it earlier. For putting something on the skin for dermatology, KPV seems like a good candidate conceptually, but we hit that barrier problem again, right? Their skin's designed to keep things out.
Absolutely. KPV, being water-loving, hydrophilic, just won't passively soak through the skin's outer layer, the stratum corneum, which is very fatty or lipid-rich. You can't just mix it into a standard cream and expect it to work underneath.
So researchers had to get creative with delivery for skin applications.
Extremely creative. They had to use pretty high-tech methods to basically force it across the barrier, things like microneedles, tiny needles that create temporary microscopic channels through the outer layer.
Ouch. Or not really.
Generally designed to be painless, just enough to bypass that very top layer. But perhaps more interestingly, they've explored using iontophoresis.
Iontophoresis, what is that exactly? It sounds electric.
It is. It uses a weak, controlled electrical current applied to the skin patch or device containing the drug, since KPV, like most peptides, carries an electrical charge.
Ah,
The electrical field helps to actively repel the charged KPV molecules, pushing them through the skin barrier and into the deeper tissues.
like using magnets to push something along.
Sort of, yeah, using electrical charge repulsion. And this technique has shown it can significantly boost the delivery of KPV into the skin in lab settings.
So that could open up possibilities for treating things like chronic wounds, maybe psoriasis, or other inflammatory skin conditions. But it requires a specialized device, not just a cream.
Precisely. The delivery system becomes just as important as the peptide itself for topical use.
Now, something else really jumped out from the research notes, this idea that KPV has a dual role, not just anti-inflammatory, but also antimicrobial. It actually kills bacteria.
Yes, this was a somewhat surprising but very significant finding. It adds another layer of potential benefit, especially in situations where inflammation and infection go hand in hand.
Like infected wounds or ulcers.
Exactly. Studies have shown KPV has direct bactericidal activity. It kills bacteria against Staphylococcus aureus.
Staph aureus, a really common and often problematic bug.
Yes, and importantly, this activity was seen even against antibiotic-resistant strains of staph, like MRSA.
Wow. So it calms the tissue, helps it repair, and fights off a superbug at the same time. That's a pretty unique combination.
It really is. It's a massive potential advantage. And it doesn't stop there. Some slightly modified versions, derivatives of KPV, have also shown antifungal activity in lab tests.
So anti-inflammatory, antibacterial, and antifungal potential, all from this tiny 3-amino acid core.
The core structure seems to hold multiple beneficial properties. This dual or even triple action makes it really appealing conceptually for complex situations like wound care.
Okay, before we get carried away with all the potential, we need to bring in a crucial finding that shows a limitation. We need to touch on the oncology research, the cancer studies.
Yes, absolutely critical to maintain balance. There were studies looking at KPV in models of colitis-associated cancer. This is where chronic inflammation in the gut eventually drives the development of tumors.
A known risk in humans with long-term IBD.
Correct. In these models, KPV did what it does best. It significantly reduced the inflammation markers in the colon. It cooled down that pro-cancerous inflammatory environment.
Okay, that sounds good.
But, and this is the key limitation, the studies clearly showed that KPV did not significantly slow down the growth of the tumors themselves.
Ah, so it treated the inflammation around the cancer, but not the cancer cells directly.
Exactly. It suggests KPV's role is really in managing the inflammatory environment, potentially reducing their risk of cancer developing due to chronic inflammation and protecting the surrounding tissue. But it doesn't seem to act as a direct anti-cancer drug that kills tumor cells.
It cleans up the neighborhood, but it doesn't take out the bad guys already established there.
That's a fair analogy. It's an important distinction about what KPV can and likely cannot do based on current evidence. It's an inflammation modulator and cytoprotector, not a cytotoxic agent. So yeah, we've painted a pretty impressive picture of the potential based on these lab and animal studies, but now we absolutely have to pivot and face the harsh realities. The gap between promising preclinical data and an actual working human therapy is often enormous.
And KPV faces some particularly tough ones, right? Starting with the most basic problem for almost any peptide drug, stability.
Mm-hmm.
We touched on it. It's just 3 amino acids long. I imagine it gets chewed up pretty quickly in the body.
Extremely quickly. It's highly vulnerable to degradation by enzymes called proteases. Our bodies are full of them. Their job is to break down proteins and peptides.
Cellular recycling crews.
Pretty much. And when they see a small unprotected peptide like KPV floating around, they basically snip it apart for parts, often within minutes.
Minutes. Wow. So if you injected it systemically, it might be gone before it even reaches the target tissue.
That's the major concern, yes. This enzyme problem means its half-life in the body is likely very short, severely limiting its ability to work unless you can get it right to the site of action very efficiently, or protect it somehow.
Which leads to the solution attempts, right?
Huh.
Trying to modify the KPV molecule itself to make it tougher.
Exactly. This involves some clever chemistry, making chemical modifications to shield it from those proteases, but it's a really delicate balancing act.
A terrifying tightrope walk, I think you called it.
It really is, because any change you make to improve stability, maybe swapping out a standard L-amino acid for an unnatural D-amino acid, or maybe bending the peptide into a ring shape, cyclizing it, could also accidentally wreck its biological activity.
You protect it, but you might stop it from being able to, say, interact with that ABI properly, or stop it getting through Pepti-1.
Precisely. You could end up with a really stable peptide that does absolutely nothing useful. The modifications must preserve that specific biological function that makes KPV work in the first place. It's a huge challenge in peptide drug design.
Okay, challenge 1, stability. Challenge 2, we keep coming back to it, delivery and permeability. Pepti-1 is great for the gut, but what about everywhere else? How do you get KPV into the brain for TBI, or deep into the skin, or maybe into joints for arthritis?
Yeah, we have to reiterate that poor permeability issue. Outside the gut, it's a major hurdle. KPV is currently best suited, based on its natural properties, for those localized uses gut, or maybe skin, with those special delivery methods. For systemic use, getting it everywhere, we are almost entirely reliant on advanced drug delivery technology.
So, beyond modifying the peptide itself, researchers are working on better packaging, like those nanoparticles you mentioned for the colon.
Yes, encapsulation is a major focus. Developing smarter nanoparticles is key. These aren't just simple bubbles. They can be designed as sophisticated delivery vehicles.
How so?
Well, they can protect the KPV from enzymes in the bloodstream. They can sometimes be targeted, maybe by adding molecules on their surface that bind specifically to inflamed tissues. And as we mentioned for the colon, they can be made environmentally sensitive.
So they only open up and release the KPV when they hit the right spot, like a pH change in an inflamed area.
Exactly. Or maybe they're designed to be broken down only by enzymes that are highly abundant specifically in inflamed tissue. The goal is targeted release, maximizing the dose where it's needed and minimizing exposure elsewhere. That's crucial for both efficacy and safety.
Which brings us to safety and specificity concerns. Okay, KPV is less likely to cause the side effects of AMSH because it avoids MC1R. But are there other risks, especially if you're using high doses or fancy delivery systems to force it into tissues?
There's always a theoretical risk, yes. Even though it's designed to be specific, we need to be sure. Besides MC1R, there are other melanocortin receptors in the body, MC2R, 3R, 4R, 5R, involved in different functions.
Right.
At the potentially high concentrations needed to overcome stability and delivery issues, researchers absolutely must confirm that KPV isn't accidentally triggering one of those receptors, which could lead to unexpected side effects we don't yet know about. Thorough off-target screening is essential.
And then there's the flip side of controlling inflammation itself. Inflammation isn't always bad, right? We need it acutely to fight infections, to start healing. What's the danger of suppressing it too much with something like KPV?
That is a critical safety consideration. Oversuppression is a real concern. If KPV is too potent or delivered too broadly or for too long, it could potentially dampen the body's necessary immune responses. You might compromise the ability to fight off a new infection, for example.
You don't want to turn off the fire alarm completely when there might still be a fire.
Exactly. The goal is modulation, calming the chronic, damaging inflammation without shutting down the essential protective immune functions. It's about finding that therapeutic window. Precision targeting helps, but it's a fine line to walk.
Okay, stability issues, delivery challenges, potential off-target effects, risk of oversuppression. All of these scientific hurdles feed into one massive practical roadblock, don't they? The data gap.
The huge lack of human data. This is probably the single biggest obstacle right now between the lab bench and the clinic.
Because everything we've discussed, the elegant mechanism, the cell protection, the antimicrobial stuff, it's all based on.
It's virtually all based on in vitro work cells in a dish and animal models, mostly rodents like mice. We have almost zero robust data from humans.
And and mice aren't just tiny humans.
Not even close, especially when it comes to metabolism, immune responses, and how diseases manifest. So we simply don't know how KPV behaves in the human body. What's the right dose? How quickly is it metabolized or cleared the pharmacokinetics? Is it safe over the long term? Does it actually work for human diseases? These are all massive unanswered questions.
So before even thinking about human trials, there's another step needed.
Absolutely. Extensive preclinical toxicology studies are required, typically in larger animal models, maybe pigs, dogs, or non-human primates whose physiology is a bit closer to ours. This is essential to establish a safe dosage range and get a better idea if the effects seen in mice actually translate. And this phase alone is lengthy, expensive, and requires specialized facilities.
And one final reality check. Manufacturing. If KPV ever did make it through all of that and became an approved drug, you'd need to make tons of it, reliably and purely.
Yes, scaling up production from lab quantities to industrial, pharmaceutical-grade manufacturing is a whole other mountain to climb. Peptides can be tricky to synthesize consistently at large scale. You need extremely high purity, guaranteed stability in the final product, and everything has to comply with very strict regulations, good manufacturing practices, or GMP.
It's not just mixing 3 amino acids in a big vat.
Not even remotely. It's a highly controlled, complex, and expensive process to ensure every single batch is safe, effective, and meets regulatory standards. That infrastructure and process development is another significant hurdle.
Okay, so we have this picture, a tiny peptide, KPV, with genuinely exciting biological potential, but it's fundamentally limited by being fragile and hard to deliver effectively outside the gut. Given these major roadblocks, what's the path forward? What are the immediate next steps researchers need to take to try and bridge this huge gap?
The research roadmap is pretty clear, and it focuses directly on tackling those core weaknesses we just discussed. Priority number 1 is chemical innovation, developing better versions of KPV.
Stabilized analogs.
Exactly. Chemists are working on creating modified KPV molecules analogs that are much more resistant to being chopped up by proteases. But again, the key is they must retain that specific NFP modulating activity. That's job 1.
And alongside making the molecule itself tougher, improving the delivery vehicles, right?
Yeah.
Getting beyond just relying on Pep-T1 or basic nanoparticles.
Absolutely. The next generation of delivery systems needs to be smarter, more targeted, and ideally responsive to the disease environment itself.
What does responsive mean in this context?
Imagine, say, a nanoparticle carrying KPV that's coated with a special polymer. This polymer is designed to be stable in normal body conditions, but gets rapidly broken down only in the specific microenvironment of chronic inflammation, maybe due to lower pH or specific enzymes found there.
So it delivers the payload right on target only when and where it's needed most.
That's the goal. Precision delivery to maximize effect and minimize potential systemic side effects. That's where a lot of nanomedicine research is heading for drugs like this.
Okay, better molecules, smarter delivery. Yeah. What else is on the critical path before humans? You mentioned larger animals and understanding the immune effects better.
Yes. Rigorous testing in those larger animal models is non-negotiable. We need solid pharmacokinetic data, how the drug is absorbed, distributed, metabolized, excreted, and extensive toxicology studies to prove safety before regulators would even consider human trials.
And the immune mapping.
We need a much more detailed picture of exactly how KPV interacts with the whole human immune system. We talked about macrophages, but what about its effects on different types of T cells, like regulatory T cells or dendritic cells? We need the full blueprint of its immunomodulatory actions to really understand its potential and risks.
So doing all that work, developing stable analogs, creating targeted delivery, doing large animal tox studies, mapping the immune effects, that's all groundwork before the ultimate goal.
That's right. If, and it's a big if, KPV or a modified version clears all those expensive preclinical hurdles, demonstrating both safety and convincing efficacy across multiple models and species.
A process that takes years and huge investment.
Years and likely hundreds of millions of dollars, yes. Then, the next step would be to apply for permission to conduct early-stage human clinical trials, phase 1 trials.
And what would those first human studies look at?
Phase 1 is primarily about safety and tolerability in healthy volunteers, or sometimes in patients, finding the maximum tolerated dose. They'd also look at pharmacokinetics in humans, how long it lasts in our system, and maybe some early exploratory biomarkers to see if it's having the expected biological effect on inflammation markers. Efficacy usually comes later in phase 2 and 3.
Okay. This whole process, the long road ahead, brings us to something absolutely crucial we need to state very clearly for anyone listening, especially given how much info and maybe misinformation is out there online about peptides like KPV. Let's talk about the regulatory status right now.
Yes. This is incredibly important. We need to be absolutely crystal clear on this point for you, the listener. Despite all the fascinating research and the potential we've discussed today, KPV is currently, strictly, an agent for research purposes only. It is not an approved drug. It is not an approved medication or treatment for any condition anywhere by any major regulatory agency like the FDA or the EMA.
That's a critical distinction. So
Yeah.
any KPV products that people might see available online or elsewhere.
Those are intended solely for laboratory research. They are absolutely not intended or approved for human consumption or therapeutic use. They haven't undergone the rigorous safety, efficacy, and quality control testing required for human medicines.
This isn't just bureaucratic red tape. It's about safety.
Right. It's fundamentally about safety and efficacy. That regulatory boundary exists for a very good reason. It underscores why all this painstaking, regulated translational research, the chemistry, the delivery work, the animal studies, the human trials, is absolutely necessary before any compound, no matter how promising an early research, can be considered safe and effective for people.
So the message is, intriguing science, potentially exciting future, but right now, firmly in the research lab, not the clinic or the supplement shelf.
Exactly. Proven safety, consistent high purity, and demonstrated human efficacy under strict regulatory oversight have to be the absolute priorities before any new compound makes that leap. The journey for KPV has really only just begun. Hashtag tag outro.
Well, this has been a really deep dive into KTV, a fascinating journey looking at, biological elegance meeting some really tough biochemical and practical challenges. Let's try and boil it down. What are the 4 main takeaways we want you to leave with today?
Okay, first, KPV is tiny. Just 3 amino acids, lysine, proline, valine, derived from the hormone MSH. Its key feature is being a potentially potent anti-inflammatory agent without the pigmenting effects of its parent molecule. It's non-melanotropic.
Right. Second takeaway. How it works is different. It seems to bypass those typical surface receptors and instead gets inside the cell to directly modulate master control switches, particularly by helping restore the natural inhibitor, IBIA, to calm down the NFE inflammatory pathway, working from the inside out.
Third, the preclinical evidence, especially in the gut where it can use the PEP-T1 transporter, is strong. It shows these protective effects calming inflammation, helping cells defend themselves, promoting repair phenotypes. Plus, that surprising bonus of having direct antimicrobial activity against bacteria and potentially fungi.
A real multitasker in the lab. And finally, the fourth takeaway. The huge hurdles. It's fragile, broken down quickly by enzymes. It struggles to get into most tissues without that PEP-T1 secret door, requiring advanced delivery tech. And critically, there's currently no significant human clinical data to confirm if it's safe or effective in people.
Yeah, it really is a classic example, isn't it? Huge promise in the basic science, running smack into the incredibly complex reality of drug development, the biochemistry, the delivery, the regulation. It makes you appreciate how hard it is to translate even a simple biological idea into a real-world therapy.
It does. And it also makes you think about that initial idea we started with, minimalist design in biology.
Yeah.
Finding these tiny ancient sequences, just 3 amino acids long, derived from our own natural proteins, acting like these incredibly potent, condensed instruction manuals for recovery and repair.
It's like we're just now learning to read these tiny, powerful blueprints that our bodies have been using all along.
Yeah. And it leaves you wondering, doesn't it? What other simple fragments, maybe overlooked bits of larger molecules are out there? What other tiny endogenous peptides hold similar potent regulatory power that we just haven't deciphered yet?
It definitely opens up a potentially elegant new avenue for drug discovery, looking for refinement and inherent simplicity within our own biology. A fascinating thought.
It really is. Something for you to mull over. If this dive sparked your interest, maybe look a bit more into how NFK signaling works or the function of that PEP-T1 transporter. They're key players in a lot of biology.
Indeed. Well, thank you for joining us today as we unpack the science, the potential, and the challenges surrounding KPV.
Thanks for joining the deep dive. We'll see you next time.